Prochnau. We have Emily PhD student into the podium.
Hello, everyone. Hello, everyone. My name is Jack Prochnau, and I am a third-year MD PhD student in Dr. Battle's lab. I'm going to present a little bit of my work today, particularly on an exciting compound I've been working on called Prochnau blue, and what I've done with it in understanding its uses to target scaffold associated regions in pancreatic cancer. Now, cancer, as I'm sure you're all aware, has been a major burden for us all. After all, it's been with us ever since we grew up and became multicellular organisms. As it stands, it is the #2 killer in the United States and 6th worldwide. In particular, and of great interest to myself, pancreatic cancer is rapidly becoming one of the most frequently diagnosed cancers. And on top of its increasing incidence, we can see from the yearly deaths below that it is outpacing itself in incidence rank by being the 3rd biggest killer among cancers. Now, this has to do with a combination of things. Of great importance is the fact that pancreatic cancer is frequently diagnosed in much later stages, with roughly 80% of all first-time diagnoses being stage 3 or higher. And even with modern surgeries, radiation, chemo, heck, even the kitchen sink, this cancer remains one of the most lethal, with 5-year survival of just 13%. So what is it that has made cancer so hard to treat? Well, I like to liken it to the fable of the Gordian knot. There has long been a legend that in the town of Gordian lied a knot tied by King Midas himself that was impossible to untie. Nothing was exposed that could be pulled on, nor a tool used to untie it. Scholars, warriors, and kings from all around came to Gordian for one reason: try to untie the knot. However, all went bad. Cancer poses such a challenge of equal complexity. How do we ultimately get rid of something that is a part of us? Well, it's these impossible challenges that has greatly interested myself and has captured the minds of many great scientists for a very long time. And as scientists, we like to tackle such challenges by poking and prodding with all the gizmos that we've developed. Also, when you just look at this thing, it even looks like a tumor. So this has led me to try to work on expanding and using our scientific toolbox in Dr. Battle's lab through carbazole blue, as shown right there. Carbazole blue was chosen among several other synthetic analogs of carbazole, which is a molecular motif used across many other therapeutics for its particular efficacy in cancer cells. This immediately prompted us to better understand why it was that CB was working so well. Chiefly, we found that CB is a molecule that has a great affinity for binding DNA, as shown by its fluorescent polarization, absorbance, and MST acids. Of interest, and that will become a bit important a bit later, is that CB also tends to have greater affinity for 18-rich DNA, as shown on the bottom right. And so, like many DNA binding agents for it, we also wanted to see what sort of kinks CB was causing in DNA-related deaths. Immediately, we found that CB was quite good at stimulating the double-stranded break response, as shown from the punctile formation with gamma H2AX and 53B1 in cancer-treated cells, CB-treated cells. Similarly, a DNA fiber assay would also demonstrate that CB could arrest DNA replication events. And of course, because I'm a very visual person, I used autodocking software to assess the possible regions of preferential binding of CB to DNA and found that CB, from the yellow at the top there, most frequently fits snugly into the minor group of DNA. So at this point, I sought to more robustly define CB in pancreatic cancer, and so ran a quick gamut of short-term viability assays to assess viability. I found that CB is effective across several PDAC cell lines, and with a respectable IC50 in the nanomolar to single micromolar range. Furthermore, I would find that CB was arresting cells in the G1 to S phase, and of course stimulating apoptosis. How about the long term? Well, similarly, CB is showing great promise in our long-term viability and migration assays. PDAC cells, even treated at sub-IC50 ranges, were clearly demonstrating static growth and low migration. Of course, the next big hurdle was seeing if CB was promising in vivo. After all, if you are killing the patient with the drug, then what such use is the drug? So I ran a quick pilot experiment using EBOCA2 pancreatic cancer cells and injected them subcutaneously into mice and treated with CB. And the results were quite promising. CB-treated mice had significantly smaller tumors, as shown in both the growth curve and tumor analysis. And what's more, is that CB was showing or demonstrating this efficacy without much change in the mice's behavior, body weight, or the liver and kidneys, demonstrating that CB was indeed therapeutically effective and safe. But now it's time to really get into the nitty-gritty. How is CB mechanistically working, and how can we better understand the molecular pathways that involve them? This led us to perform chemical sequencing, or ChemSeq. This function is akin to chip sequencing, where we biotinnate our CB, treated cells, and then pull down the DNA we bind to. And what was particularly interesting was that the analysis of this data demonstrated to us that CB was binding to regions of DNA that SAP1 and HMGA1 bind to. These proteins are scaffold matrix associated regions, SMAR, associated proteins, that have a wide range of activities ranging from gene expression to chromatin structure. So what the heck are SMARs? Well, SMARs have both genetic and epigenetic roles, but their role is primarily epigenetic. They are 18-rich DNA sequences that anchor chromatin to the nuclear matrix, helping to define chromatin domains and influencing overall gene expression. Epigenetically, they regulate chromatin accessibility, enhance motor interactions, and replication timing, among other vital functions. SMARs are therefore integral, higher order DNA structure and gene expression, and are frequently dysregulated in cancers of all kinds. And we see that if we go back to our previous slide, we can see that SMAR-related proteins like HMGA1 are largely associated with poor patient outcomes, indicating an oncogenetic role. But when CB is binding to these regions, such as the cyclin E2 promoter that HMGA1 associates with, it is effectively shutting down transcription and ultimately translation of these genes. Now, this is one current arm of my project, and I hope to better understand how SMARs and SMAR-related proteins are involved in oncogenetic processes. However, there is one more arm of the story that I wish to tell. Namely, when I performed RNA sequencing on CB-treated cells, not only does a robust activation of apoptotic processes, as shown in the above graph, but of very great interest was that I saw an enormous downregulation of gene products associated with splicing, as shown in the red boxes. Many of the most downregulated genes were part of the U1 small nuclear RNA family, or snRNAs. Now, snRNAs are a required component of small nuclear ribonucleoproteins, or SNRs, for proper spliceosomal assembly. Without these critical RNAs, RNA splicing is globally repressed, leading to leakage of improperly spliced RNA into the cytoplasm. And once in the cytoplasm, these RNAs will be translated by ribosomes into non-functional peptides that will go on to bloat the endoplasmic reticulum in an attempt to properly fold and process them, leading to an unfolded protein response, or UPR, and inevitably leading to apoptosis. And indeed, if we go back to our gene enrichments, we can see that cells treated with CB are undergoing a robust unfolded protein response and ultimately engaging in apoptosis. I'll of course squeeze in just a bit more of my most recent preliminary data, confirming these RNA-seq results where I am seeing transcriptional loss of U1 snRNAs and a significant increase in gene expression of proteins involved in the UPR response, such as XBP1, CHOP, EIF2 alpha, and phosphopyridine. So here's where I'm at. CB, with its particular avidity for 18-rich regions of DNA such as SMARs, is greatly disrupting oncological processes in a cancer cell, causing genome instability and DNA damage, all whilst having another unique arm, where it is draining the cancer cell of snRNAs needed for it to drive its rapid growth and resulting in a drastic UPR response. Now, if and exactly how these two processes, the SMAR and U1 snRNA depletion, are linked, is something I'm interested in exploring. But that is for future presentation. Now, I want to take a brief final moment to truly emphasize the incredible progress we've made in the past 50 years. We've put some serious elbow grease into cancer, and some may now say that we have plucked all of the low-hanging fruits and that it'll be agonizingly slow in our commitment. But I say that this era will mark for an unprecedented growth in our understanding of cancer and how to treat it. Much like the disease we are fighting, I remain aggressively optimistic that we will hopefully be putting ourselves out of a job within the century. Of course, I thought it would be a little fun to add ChatGPT's rendering of cancer as a metaphorical modern day word. So I'd just briefly like to acknowledge my mentor, Dr. Manjeet Rao, who's been incredibly supportive, and of course all of the remaining lab members who've been extremely helpful. Nir, Boston, Suba, Deepika, Santosh, Daisy, Pat, Zina, and Shahab, along with our collaborators, the funding I received, and the support I've gotten from the medical scientist training program. With that, I will take any questions.
How do you envision translating this one to pancreatitis?
Yeah, so I would never imagine, especially in the modern day, that CB is going to ever be its own monotherapy. However, I believe that it will replace or at least better the treatment plans that we do have for pancreatic cancer. One of the biggest problems that I didn't address as a point explicitly is that the treatments for pancreatic cancer, such as modified FOLFIRINOX treatments, so formic acid, oxaliplatin, these are incredibly toxic treatments. And furthermore, the people who we are treating for pancreatic cancer are usually 70 years old or older. So usually they can't tolerate this therapy. So I envision that CB as a relatively potent therapeutic has the safety efficacy profile can lower this burden of morbidity of the treatment that we have with pancreatic cancer. Furthermore, the really unique aspect of CB about binding these interior regions like SMARS is of really great interest. So really I want to investigate further how these SMARS, scaffold matrix associated regions, are implicated in pancreatic cancer along with all of the other cancers that we have.
Thank you. Well, I was just going to ask you about something similar, and that is what is the kinetics or the pharmacokinetics of this medication. How do you envision perhaps to do it, in situ, targeting explicitly, the cancer cells or the pancreas, bypassing all the other organs? What are your thoughts? What is known about this molecule in terms of delivery?
Yes. PK studies done previously demonstrated that CB is generally cleared with a half-life about 16 hours. Again, it's just a broad-based chemotherapy, so it is going to just, once you inject it, it's going to go everywhere in the body. And every cell in the body has SMARS. However, much like how cancers abuse and use different genetic pathways to, either provide the means it needs to grow or divide, if you can shut down those mechanisms, you can either make them static or make apoptosis. That's ultimately what we're hoping that CB is going to do. This isn't going to avoid the main issue of the fact that this is still going everywhere. So I'm sure down the road, would this go to clinical trial, we'll probably see some kind of hepatotoxicity or nephrotoxicity, but at least preliminary experiments demonstrate that it's powerful. Any further questions? Okay, all right. Oh, I'm sorry.
Is there a reason why, just on top of just a brief presentation, is there rationale why CB is more equivalent to the minor groups versus the major groups of DNA?
So that's a really good question. In terms of just the general lowest energy state, that's what I was generally getting when I was running these auto development programs with CB against DNA. Why it is exactly that it's preferentially binding the minor group, I guess it's just a matter of sterics and accommodation of the molecule, such that it fits more snugly into that center and stays interpolated. But it's very likely that CB, of course, it's not going to just only go into minor groups, it's going to be equally go into, well, not equally, but it's going to also interpolate into major groups as well. But the fact that it fits so snugly into that minor group can indicate that what's in there is insane.
Thank you.
The next presenter is from the graduate student. And I welcome Ernie Hopper, up to the microphone, please.